this presentation, a lot of the slides, if not most of them, are in French. And I will speak mostly in English, but I will also speak in French at times when I think that maybe the biology, some aspects of the life history of these animals can be maybe mentioned in French as well. So this is a topic that I've been covering for the past, for the past couple decades. And freshwater mussels, you've seen them. And you've seen them when you go canoeing, you've seen them at the lake, and you've seen them in rivers, Ottawa River and elsewhere, regardless of the province where you go, except in the highest latitude in the Northwest Territories. They're present from coast to coast, inland, in freshwater environments. So today, what I would like to do is to talk about this fauna, which is really extraordinary in a way. We've seen them, we see them all the time, but their life history is by far rather obscure and unknown to most people, the details of which, when you start digging and when scratching the surface, really is fascinating. Donc, ce que j'aimerais aujourd'hui, c'est parler de cette faune des moules d'eau douce, mais avant que j'attaque le sujet, si vous voulez, la faune de moules d'eau douce du Parc de la Gatineau, entre autres. Juste quelques mots sur l'endroit où je travaille. Moi et mes collègues au Musée de la Nature, vous connaissez le Musée de la Nature au centre-ville d'Ottawa, les expositions nationales. You know about the VMB downtown Ottawa, but you probably also know the Natural History Campus, the NHC, on Pink Road in Andrew. This is where all the national collections, all the natural history collections of the Museum of Nature are located. These are the best, largest, oldest natural history collections in the country. This has been, in the making since the mid-1800s. And on Pink Road, it's about, the size of a football field where all those collections are found. So in this building here in Gatineau on Pink Road, you have about 15 million specimens. Can you imagine? So my colleagues, myself, we work on a variety of faunas, floras. And if you look here on this side of the screen, from crustaceans, echinoderms, mollusks, insect, fishes, the faunas and floras of Canada, that's the best place to study biodiversity. And we have all these records available as well. So these are unequal collections by far, and that's one of the strengths of the Museum of Nature here in Gatineau and Ottawa, to show and to study these specimens. And for me, what's fascinating is to, of course, study biodiversity. If I just open a few drawers, this picture was taken by my colleague Jean-Marc Gagnon. Sur cette photo, on peut voir, là, vous avez une idée, lorsqu'on se met à ouvrir les tiroirs, de ce que vous pouvez voir dans ces cabinets. À droite, là, j'ai ici je pointe avec une flèche rouge. Ça vous donne une idée de la collection de moules d'eau douce. On a 70 cabinets d'environ 1 mètre cube. C'est la plus grande collection nationale de moules d'eau douce. De Terre-Neuve aux îles Charlotte, Idaho, Yukon, j'en passe, et à travers le monde. Mais pour les moules d'eau douce, c'est surtout une collection canadienne, ce qui la rend très précieuse. Et puis, ça vous donne une idée là, de l'ampleur de ces collections. An example of what you could do with these collections, if you just look at these specimens, these records, have a look at the upper left. So this is the dwarf wedge mussel, la lastidonte naine. That's the first officially extirpated invertebrate in Canada. It was on the way down when they started to build a dam along the Petit Kodiak River in New Brunswick. And we have a record, the collections. We have records of this species in New Brunswick. It was only found there at the time, prior to the construction of this dike on the Petit Kodiak River near Moncton and Riverview in Moncton, near Moncton. So the point is, these collections allow us to go back in time and study biodiversity through time. Bottom left, should I say? Yeah, bottom left. One of the oldest records of the hickory nut mussel. Extirpated from the Great Lakes. Reason? Zebra mussel. La moule zébrée a été introduite. Il y a aussi eu un déclin de l'esturgeon jaune, the decline of the lake sturgeon, which is its host fish. In the Great Lakes, the sturgeon have declined tremendously, and because of the zebra mussel, many freshwater mussel species are extirpated from the Great Lakes. And that's the case of this one right here, the pink hill splitter, le potamille eri. Anyway, the reason I'm showing this to you is to give you an idea of the usefulness of such natural history collections. The one of the Museum of Nature is hands down the best in the country. It's our national collection of natural history. And we can go back in time and determine how the environment has changed. Fishes, plants, mammals, birds. So freshwater mussels, in my case, is something that I'm interested in, and I've tried to convince you why. They are an incredible, it is an incredible fauna. So the plan of the talk today, number one, qu'est-ce qu'une moule d'eau douce? What's a freshwater mussel? You guys probably know some of it, but I'll cover some items that might surprise you. I'll talk about the freshwater mussel fauna of the Outaouais region of the Ottawa River watershed. It's extraordinary. And you'll see why we need to protect the Ottawa River watershed, not only for its water quality, but also for the fauna that you find there. The fishes, the mussels, the biology, an amazing life history that these mussels have. I'll try to cover that very rapidly. I have many underwater photographs, and I'll talk also about what do they do in the ecosystem? What's their ecological function? I'll talk about conservation also, and then I'll focus the remaining half of the talk, or 1/3 of the talk, is about Gatineau Park, the kinds of studies that myself, colleagues, volunteers, university students we've done and we continue to do in Gatineau Park. Donc voilà les 4 objectifs de ma présentation. Alors si on commençait, on va aller tantôt à l'ITM, mais avant, mon but premier pour cette présentation, vraiment, c'est qu'à la fin de la présentation, lorsque vous aurez terminé, lorsque j'aurai terminé de réaliser ma présentation, c'est de vous faire voir les moules d'eau douce sous un angle différent, parce qu'elles sont vraiment uniques en termes de biologie. Et la relation poisson-moule est fascinante. Et c'est de vous convaincre de cette connectivité qu'il y a entre les moules et tout le reste de l'écosystème. So at the end of the presentation, that's my goal. I'd like you to see freshwater mussels in a different way, and how connected this group of animals in our lakes and rivers, really, how they are connected to the rest of the ecosystem. All right, first objective. What is a freshwater mussel? So don't mix them up with the zebra mussel, which is a totally different systematic or taxonomical group. The zebra mussel is not what I'm talking about today, although I'll slip in a few words about them. They're introduced, they crossed the Atlantic, and they did cross the Atlantic via ballast water. It's a freshwater organism, and I'm not talking about them in details today, although I'll mention a few things. Don't confuse our native freshwater mussels with, quote, the edible mussels, which are marine, which we eat at the restaurant or can buy at the seafood market. So I'm going to talk about those mussels, which you do know. You've seen them before, and they belong to an order called Unionida or Unioniform. Union and mussel is another way to express or tell them. This is beautiful material, folks. Look at this. These are underwater photos I took here of pearl shells. They're found in West Coast rivers, East Coast rivers, and one of them is right here near Montebello at Canuck. So here's the eastern pearl shell. It's beautiful. And look at the interior of some of these species. They're all nacre, shiny, pearl-like inside. Often they're called pearl shells. All our native freshwater mussels have a pearly inside of their shell. The outside is typically dark, brownish, and shows growth rings. Du beau matériel, c'est clair. Regardez en passant les perles. Je n'irai pas en détail, mais certaines espèces sécrètent des perles. n'ayez pas l'intention d'aller les rechercher pour les perles, elles sont protégées par la loi. Toutes les moules d'eau douce sont protégées par la loi. Une moule d'eau douce, c'est un animal vivant complexe. Many of you are aware of it, but I just want to touch base on a few items. These are mollusks where the body, the soft body, is enclosed between two valves. That's why we call them bivalves. They are with two powerful adductor muscles closing the valves together. They have a foot, powerful muscular foot, allows them to dig into the sediment and sometimes even move horizontally on the sand or silt. They have a mantle, siphonal apertures right here. This is where the water comes in. This is where the water comes out. And these are the gills. They have a heart, digestive system. They're typical filter feeders, like many, quote, marine bivalves, like the clams and the oysters and the scallops in the ocean. Our native freshwater mussels do the same kind of thing, but in our lakes and our rivers. Donc, ne pas oublier qu'une moule d'eau douce, c'est un animal complexe, vivant, qui a tous les organes nécessaires pour vivre dans nos rivières et nos lacs. Le pied musculaire, here's the muscular foot. Many of you folks, I'm sure, you've seen this before. You go canoeing or you hike along the lake shore, you walk in shallow water, you can see these tracks. You've seen them making these tracks. Sometimes they can plow like this for tens of centimeters, sometimes a meter in a day. If the sand is soft, the silt is soft, and there are no rocks, no obstacles. So they're known to have a powerful foot. Here's a juvenile that this larger adult mussel is pushed out of the sediment. I photographed this in a river in Quebec. With this picture, what's my intention? No doubt, I want to impress you by how complex, how beautiful the mantle, the siphons really are for so many of these species. Here's the shell right here. A very good close-up there with my micro lens. I'm very close to it. Here's a large, 10 centimeter mussel, and we can see where the water comes in. This is the incurrent siphon where the water comes in. Look at the complex papillae of this species. I took this picture in the Quinonge River right here, near, not too far from here, in near Montebello, in the Canuck forest, at the Canuck Forest Institute. The color, the papillae, each species has its own signature. So if you have a good macro lens, a good camera, underwater photo is easy these days. Portable cameras, cell phone can be put in a Ziploc. Take a picture of the siphonal apertures of an undisturbed mussel, and if the picture is pretty good, send it to me. And I likely will be able to identify it without you to do anything but just take a picture. So here are some examples. This one here on the upper right is the Eastern Elyptia. Cette espèce-là, c'est l'Elyptia de l'Est. C'est le plus commun, ou la plus commune des espèces dans l'Est du Canada. Et on retrouve ici encore celle-là au Parc de Gatineau. La lame cédrier, aussi retrouvée au Parc. Donc, deux espèces, ici une et celle-là. Bref, cette photo, c'est pour vous donner une idée que les siphons, l'animal, lorsqu'il n'est pas dérangé, c'est complexe, il y a des papilles et chaque espèce a une signature ou sa propre morphologie, ses propres couleurs. Comment est-ce qu'on identifie les moules d'eau douce? Très facile. En bref, les caractères sont assez simples, mais ça prend quand même de l'expérience. So, briefly, check the shape, check for growth rings, rays on the shell, check for hinge teeth, the color inside, the color outside, the position of the beak or umbo. So each species has its own, morphological traits. Donc, c'est pas tellement difficile avec le temps, on s'habitue à identifier assez facilement les espèces les plus communes. Regardez comme il faut ici. On va voir des dents. These are hinge teeth. So imagine a kind of a jewelry box. The two hinges, the hinge allows the two valves to stay aligned. So these are hinge teeth right there. Here's one, and here's another one. By the way, these are where the muscles are located to close the animal. By the way, of course, this is a dead shell, an empty shell, and I'm showing you this to give you an idea of how to identify the species looking at empty shells. Each form, each color, each shape means one species. So with experience, You can, you know, tell them apart. Here's an example. Hickory nut mussel. This is a collection that myself as a diver and volunteer divers we did in the Ottawa River, not too far from here. And hickory nut, one species is here, the plain pocketbook. Another one here found in Gatineau Park. The fat moquette, one here à la Spidonte à Fortedam. Triangle floater, also found in Gatineau Park. Here's the most common species in Gatineau Park and Eastern Canada, widespread in our lakes and rivers, the Eastern Elliptio. So each species is quite easy to tell apart from this picture, just looking at the shapes, color, rays. How many species are there? Quickly, 900 species of freshwater mussels belonging to this large order in the world, 900 species. Canada has 55. It's not bad. 6% of all the freshwater mussels of the world we have. Look at Europe. Europe, all the countries of Europe, they have 12 only. Wow. North America and Southeast Asia, the two richest places on the planet for freshwater mussels, with around 290, 310. But Canada, even though we're a northern country, it's pretty good. Sorry for the rain. Here is on item 2. Let's talk about the richness of the Ottawa River in terms of mussels. And you'll be amazed. It's a large river. You know this river. It's wide, high flow, powerful. It's a vast system. And look at the size of this. It's 1.8 times Lake Superior. Wow. It is 2 times the surface of New Brunswick. That is how big the Ottawa River watershed is. It's extraordinary. Look at the flow. Mean flow is about 5,000 to 7,000 cubic meter a second at Carillon near Montreal on a normal spring year, on a normal April and May. But look at the 2 floods we've had, by the way. We reached above 9,000 cubic meter a second in 2019 May. This was a discharge that was larger than that of the entire Great Lakes at Cornwall. That's extraordinary. One key element, good news, the upper portion of all of the Ottawa River watershed has no zebra mussels, especially on the Quebec side. Zebra mussels are only present when calcium content, when the water is hard enough with calcium in it, dissolved calcium, to reproduce. So they are present, these zebra mussels, an invasive species, more like between Ottawa and Montreal, but the impact is modest. It depends where you are. It is more serious on the Ontario side and some of the tributaries. Anyway, the point I want to make with this picture, 35% of all the freshwater mussels of Canada are found right here in our region. And when people see this, they're amazed. They're surprised. So this is the list of species of freshwater mussels that you have right here in our watershed. Gatineau Park mussels found within the territory of the park. They're in green. So it's not bad. You can tell that Gatineau Park has an interesting diversity of freshwater mussels. This one here is an endangered species, and I'm not going to talk about this one. It's the subject of another talk. It's linked to the sturgeon, and we studied it in the Upper Ottawa. The point of this slide is this also. Some of these sites along the Ottawa River watershed have about 100, 200 large freshwater mussels per square meter. That is an incredible density. It's actually an incredible biomass. In some areas, the density of mussels can be extraordinary. So watershed, this watershed of ours has 38% of all the species of freshwater mussels found in Canada. That's extraordinary. So 38%, it depends on 35, 38% of all the mussels of Canada are here in our watershed. These are some examples. The ones in red color, we have them in Gatineau Park. You can see how different they are in appearance. Look at the growth rings, shape, position, color, striation, ridges, here rays, and here you've got some of deep gouge type ridges at the posterior end. That's where the umbo is, or beak. This is the beak right here. That's anterior, and that is posterior. The siphons are always at the posterior end. The foot and how they bury is always from the anterior end. So anterior and posterior. Anterior, posterior. This one here is oriented differently. So here's the umbo. Near the umbo is anterior where the foot is and posterior where the siphons are coming out. Item 3. Let's talk about biology, conservation, ecosystem services. That's where it becomes interesting. They're filter feeders. So that, I've mentioned that earlier. They are equipped with gills. They filter the water of our rivers, lakes. L'espèce que vous voyez ici, c'est l'élétio de l'Est. Je le mentionne parce que c'est l'espèce la plus commune, la plus abondante, souvent même par mètre carré de fond de rivière ou de fond de lac, partout dans l'Est du pays. Et le parc de la Gatineau n'est pas une exception. Ce que j'aime avec cette photo que j'ai prise dans le secteur, encore une fois, de Québec, regardez les branchies. J'aurais pu la prendre dans la plupart des lacs du parc de la Gatineau. On voit très bien les branchies qui vont capter le plancton. Ça, c'est l'ouverture inhalante par où entre l'eau. On voit les belles papilles que je vous mentionnais plus tôt. Le siphon ou l'ouverture exhalante par où sort l'eau. L'eau entre par ici. L'animal capture à l'aide de millions de petits cils vibratiles les particules planctoniques, les retient, les achemine à la bouche, ça rentre à l'estomac, on mange et parfois on rejette évidemment des petites particules qu'on ne veut pas ingérer et de même que les excréments qui sortent par le siphon ou l'ouverture exhalante. Un exemple de plancton, de diatomées, de petites algues planctoniques microscopiques, ici en haut, qui sont la nourriture de ces moules d'eau douce. Alors, ce qui est important aussi de mentionner, en passant ici en bas à droite, en bas à gauche, la biomasse des moules d'eau douce, normalement, en termes de poids, ça représente 90, parfois 99% de la biomasse par mètre carré de fond de lac ou de rivière. C'est une biomasse qui est dominante, hands down, comme on dit, en bon canadien anglais. Alors, cette photo-là vous montre un autre exemple d'une moule qui filtre l'eau. Alors, elle est en position de filtration. L'eau entre par ici. J'ai pris cette photo-là dans une rivière pas trop loin d'ici. L'eau entre par le siphon inhalant et sort par le siphon ou l'ouverture exhalante. Encore une fois, le plancton. Regardez les volumes d'eau dont on parle. On parle qu'elles sont filtreurs. So imagine this. Imagine you filter 2 liters per hour. One large adult mussel can do that. So that means what? You're filtering 80, should I say 48. Around 40, 48 liters of water is filtered each day. One mussel. That is thousands of liters per year. One mussel. Imagine a lake, a river with millions and millions of mussels. It's extraordinary. What a service they provide. And they'll do it for 10, 20, 30 years. Some species can live 100 years. So the filtration capacity of our native freshwater mussels must not be taken lightly or must not be forgotten. It's an extraordinary service. I mentioned here one thing. Look at the upper right corner of the slide. Coliform fecal. Yes, native freshwater mussels can eat coliform bacteria and digest them. So they do eat bacteria, detritus, macroscopic algae, and they are obviously able to do other things than just filter feeding. You have seen these tracks before, I'm sure, many of you canoeing, exploring. They are able to move across the sand, mud. It's time to overwinter. Some of them will bury in the sediment, disappearing just a bit. So they're able to mix the sediment of our rivers, of our lakes, and this oxygenation process, you could call it the earthworm effect. And it's a great service. They are mixing up oxygen. They're mixing up the substrate, the bottom of the stream, the bottom of the river, as long as there are not too many cobbles, gravelly type substrates, rocks. Donc, cet effet-là, j'aimerais le souligner davantage, l'effet vers de terre. Et il n'a pas tellement été quantifié dans la littérature scientifique, mais on sait que ça se passe. Il y a beaucoup de mélange de sédiments, et ça, c'est merci, en fait, grâce au pied, le pied musculaire qui est enfoui dans les sédiments. Donc ici, on voit la portion postérieure. L'animal est partiellement enfoui ici. Je l'ai dérangé un peu lorsque je plongeais. Il est fermé, mais lorsqu'il est ouvert, il va être capable, avec son pied, de ramper par reptation. Il se déplace et on voit le sillon qui est laissé derrière. Et je mentionne ici, ça c'est un système, c'est un comportement, c'est une fonction écosystémique que la moule zébrée envahissante ne nous donne pas. Okay? La moule zébrée rend le fond anoxique. Nos moules sauvages oxygènent le fond de nos lacs. Ou font un apport d'oxygène, si je me reprends ici. Vous les avez vues souvent mortes sur le bord du lac, sur le bord des ruisseaux. Évidemment, les moules d'eau douce servent de nourriture aux rats musqués, aux loutres de rivière, aux visons, aux oiseaux, et j'en passe. Ratons laveurs, le prédateur numéro 1, et c'est le cas au Parc de la Gatineau, lac La Pêche, lac Philippe, vous allez voir ça, c'est le rat musqué. Alors souvent, on voit ce qu'on appelle en anglais des muskrat shell midden. Donc, what is a muskrat shell midden? Typically, it is a pile of shells that the muskrat has left after eating the freshwater mussels that the animal has, taken from the bottom, brought them near shore, come out of the water, nibble the edge of the shell, crack just a tiny bit of it. Then the mussel will lose its water tightness, if you want, its mantle connection with the valves, and that will lead to dehydration. The muskrat will come after a few hours, a day or so, and the freshwater mussel is dead or dying. The valves are gaping, and then the muskrat will eat the inside. So you've seen that before. Point is, food for wildlife. That's an important service that they provide. Now, this is a key slide for my presentation. So pay attention to what you've got on your screen. Cette diapo-là, je vais passer une bonne minute ou deux à l'expliquer parce qu'elle est cruciale pour comprendre que font nos moules d'eau douce dans nos lacs, dans nos rivières. Partout au pays, c'est le même scénario. So everywhere you go, if you see freshwater mussels, from British Columbia, rivers to lakes and rivers of Newfoundland, they are linked to fishes. If there are no fishes around, no local freshwater fishes, from minnows, bass, stickerbacks, then there's no reproduction of freshwater mussels that's possible. So they are entirely dependent on fishes to make juveniles, to reproduce. So here's a male, let's say, releasing the sperm at the time of spawning. The female, as the female is filter feeding the plankton, the sperm will get into the female shell, live shell. Of course, these are live animals at this point. And the sperm will fertilize the eggs that are brooded into specialized compartments that we call marsupial pouches. When I say marsupial pouches, you will think kangaroo. Well, the gills have specialized pouches where, in addition to capturing the plankton, there's a little place where the eggs are kept, developing into Glochidia larvae, which is a new word for you maybe. These are specialized larvae that are kept into the female, inside the female, being brooded. When the right fish is nearby, and that's an aspect of biology that we know little, when the fish are nearby, very close to the freshwater mussel, the female will release the Glochidia by clouds of hundreds of thousands of tiny, tiny Glochidia larvae. They are usually between 0.1 and 0.3 millimeter in size. That's microscopic, smaller than a grain of sand. Les Glochidies, une fois relâchées, souvent c'est été, automne, pour cette espèce-là ici, en passant c'est l'automne, la Glochidie doit s'attacher soit aux nageoires du poisson ou aux branchies, ça dépend de l'espèce. Pour cette espèce-là, pour l'anodonte que j'ai ici sur cette photo, c'est les nageoires. Les différentes nageoires vont faire l'affaire. Plusieurs espèces différentes de celles-là, ce sera les Glochidies, devrais-je dire, vont aller s'attacher sur les branchies du poisson, derrière l'opercule ici. Donc, c'est en respirant avec la bouche, en ingérant l'eau par la bouche et respirant, que le poisson va ingérer ses larves ou Glochidies de moule d'eau douce. C'est tout le temps comme ça que ça se passe. soit les nageoires, typiquement, ou les branchies, dépendamment de l'espèce de moule d'eau douce. Après avoir passé X nombre de jours, semaines, parfois des mois, dépendamment de la température, il y a un relâchement, une relâche des embryons, des moules qui se sont métamorphosés sur le poisson et la moule est maintenant une petite moule juvénile de moins d'1 millimètre et ça prend environ 2 ans avant d'émerger et qu'on puisse la voir à l'œil nu sur le fond du lac. So it's an amazing life cycle. And as I mentioned earlier, after attaching on the fins or the gills, the larvae, spending weeks, sometimes several months on the fish, will drop off when they have metamorphosed, when the inside organs of the animal have completed their differentiation, when they have a little mini stomach, mini heart, mini gills, all the equipment, all the necessary organs to make a living as a tiny mussel. They'll stay after dropping off from the fish 1 to 2 years out of sight. They're so tiny. After a year or two, they will emerge at the surface of the bottom of the lake or the river, and then they'll take years, normally 4, 5, 6, 7 years before they start reproducing. Some will not reproduce before 10 years old. So these are long-lived animals. They have a complex life cycle. Let's call that sophisticated life cycle, thank you. They require fishes. No fishes, no reproduction. So it is the fishes that disperse all the freshwater mussels you see in a lake or a stream. There would be no other way to go against the current and colonize a brand new river, especially at the end of the Ice Age. You needed the fish going up the rivers, estuarine fishes and whatnot, freshwater fishes, to come up again the river and the lakes to colonize the lakes and the rivers of mussels. So they are indispensable in their reproduction. This complex life cycle made the mussels very vulnerable to, quote, environmental change. Whatever you do to the fish, and each mussel will have its own 1 or 2 or 3 fish species, normally several, that can allow this dispersal and this metamorphosis. Not every fish will do. Each species of freshwater mussel has its own set of fish species that are required. Donc, c'est un cycle vital complexe qui nécessite des poissons spécifiques pour chacune des espèces de moules d'eau douce. Alors voilà, si vous avez compris en bref cette diapo-là, vous comprenez un peu ce que je vais mentionner un peu plus tard sur la connectivité et pourquoi les moules sont d'excellents indicateurs écologiques. Un exemple de larve. Alors, ici, vous voyez une larve de l'espèce Anodonte de l'Ouest. On voit ici les 2 petites larves attachées au bout de la nageoire d'un poisson, fortement grossi avec un microscope. On voit les rayons de la nageoire. On voit que les 2 flèches ici, les 2 petites larves, les 2 petites glochidies. These 2 glochidia, about 0.3 millimeter, 1/3 of a millimeter, if that. And coming out after metamorphosis, they drop off on the bottom, and here's a tiny, tiny 1/2 millimeter, 0.4 millimeter, that's 200 micron, 0.2 millimeter. So this one has a foot, this one is metamorphosed, this one is ready to go. It's ready to start a new life on the bottom of the fish as a tiny, tiny mussel. So I took these pictures at the Museum of Nature in the lab. So on this picture here, I'm going to show you a video, and look at this. You're going to see the various stages I just showed you earlier on the previous slide. Very quickly, inside the female, here they are clamping on. We're looking for a fish. Here they are. You can see them, they're clamping, they're looking for a fish. The female will release them at the right time. They will have a tiny adductor muscle that will allow the valve to close. Here you see them on this video attached to the edge of a fin of a fish, and here's you've got the juvenile that came off metamorphosed. So if I repeat this video, here we are, the mass of glucidia inside the brooding pouches of the female. You can see them, they're alive, they have a tiny adductor muscle, these specialized larvae, they want to hang on a fish. So when they will be released by the female, with this tiny adductor muscle, which you can see right here between the valves, they will attach on the fin of the fish or the gill of the fish, depending on the mussel species. Here they are. If I stop here, you can see here's the fin of the fish, highly magnified, and they're pinching and, attached just at the edge. They'll stay there for weeks, if not months. Here's the adductor muscle. On voit le muscle adducteur qui permet à la moule de s'attacher et de fermer fort contre la nageoire ou la branchie du poisson pour un certain temps, le temps de la métamorphose et de la dispersion. Ça, c'est une publication pour cette espèce-là qu'on a publiée éventuellement pour décrire ce qui se passe chez cet anodonte. Alors, OK, prochaine photo. Très brièvement sur ce sujet, mais il est trop fascinant, je n'ai pas pu m'empêcher. Certaines espèces de moules d'eau douce sont capables d'attirer le poisson pour maximiser la possibilité, la probabilité d'avoir les toutes petites larves s'attacher avec succès sur le poisson. Ici, c'est une photo que j'ai prise dans la rivière des Outaouais. Au parc de Gatineau, il y a une espèce qui lui ressemble à ça, mais elle n'est pas exactement comme ça. Qu'est-ce que vous voyez sur cette photo? Une moule femelle, enfouie presque entièrement, pris avec une lentille macro, et on voit ce qui ressemble à un petit poisson, mais ce n'est pas un poisson. C'est la chair de la moule femelle qui, lorsque érigée et en mouvement, va imiter un petit poisson qui va attirer un achigan, une perche chaude ou un poisson, qui va vouloir mordiller cette simili-appât. Et à ce moment-là, la femelle va relâcher ses embryons. It's an incredible way to attract a fish and to increase the proximity between mussel and fish. So some species, not all, are able to imitate a bait that the fish most of the time cannot resist. This is the plain pocketbook, La Lampsile cordiforme, a picture I took in the Ottawa River. By the way, this picture was taken near Britannia Bay. That's not very far from here, folks. Right here near, near Ottawa, off Britannia Bay. So here you've got this kind of a minnow imitation. You can even see it has the imitation of an eye spot. Here's the siphon. Here are the siphons, by the way, of the mussel, female. And here's the tail of this fish-looking thing, and here are some spines. And it pulses every now and then. It is an incredible device. How to do survey? A few words about methods. Well, look at this here. You can use an aquascope, which allows you to see the bottom quite well. You can use scuba diving, which we have to when it's too deep. Or you can use a wetsuit, a quadrat, and count mussels, describe the community, identify the species on site or bring some to the lab. With a keen eye, you can, identify each species quite easily with practice. Donc, on voit ici un peu les différentes méthodes qu'on peut utiliser pour l'étude des moules d'eau douce. Alors, quadrat, aquascope, scuba. Ici, on peut voir là, un peu la méthodologie. Rapidement, j'avance ici vous mentionnant que la conservation des moules d'eau douce, c'est un problème majeur. On peut voir ici que parmi les espèces, les faunes, les groupes fauniques les plus en déclin en Amérique du Nord, les reptiles, ça ne va pas très bien. 80% ou à peu près des espèces de reptiles de l'Amérique du Nord sont en déclin, en péril, espèces menacées. Ça ne va pas très bien pour les reptiles, si on le savait. Mais regardez ces 4 groupes d'animaux aquatiques qui vivent dans les lacs et les rivières de l'Amérique du Nord. Moules d'eau douce, 72% des espèces sont en péril ou en déclin. Wow! Les écrevisses, on sait qu'il y a beaucoup d'espèces aux États-Unis, d'écrevisses en déclin. Les amphibiens, vous les connaissez. Les poissons d'eau douce. Ces 4 groupes-là, on peut voir que nos eaux douces, il y a bien des endroits à travers l'Amérique du Nord où ça ne va pas très bien pour plusieurs espèces. Comparativement à seulement 14%, peut-être que mon chiffre ici pourrait être révisé à 16%, des oiseaux de l'Amérique du Nord qui sont en déclin, devrais-je dire en péril en tant qu'espèce. Bref, au Canada, ce que j'aimerais mentionner ici, si la valeur est de 72% pour les États-Unis et l'Amérique du Nord en général, elle est 35% de toutes nos moules d'eau douce au Canada qui sont à risque. Donc, retenez qu'environ le tiers de toutes les espèces de moules d'eau douce du Canada sont en déclin, en péril. So, about 1/3 of all the freshwater mussels of Canada are at risk in one category or another. Zebra mussel, just a few words. We know it's a pest. It crossed the Atlantic in our large cargo ships. It lives in freshwater. It was introduced in the Great Lakes. It has caused havoc out there. it has meant the extirpation of so many millions and millions of freshwater mussels in the Great Lakes. Most species of the Great Lakes are now gone. There are some pockets in refugia, but the zebra mussel, wow, what a problem we have. It is not, however, in Gatineau Park. That's the good news I want to slip in. The zebra mussel is not in Gatineau Park lakes, not now. And hopefully it will stay that way. This is a picture just to show you. Before the invasion of the zebra mussel and after. On peut voir l'impact absolument incroyable que la moule zébrée a sur les espèces de moules d'eau douce sauvages, indigènes. Ici, là, la mortalité va être de 100%. Il n'y a aucune chance de survivre lorsqu'il y a autant de moules zébrées attachées à une pauvre moule d'eau douce comme ça ici. On parle parfois de centaines de moules zébrées attachées. L'achigan, j'en parlerai pas cette fois-ci, mais bref, introduire un poisson qui n'appartient pas à nos lacs, c'est aussi un problème. Je n'irai pas dans les détails, mais indirectement, ça va affecter la reproduction de nos moules d'eau douce avec les poissons indigènes qui sont malheureusement mangés par cet achigan qui n'appartenait pas à nos lacs dans les siècles passés. Clairement, les moules d'eau douce, un peu avec ce que je vous ai dit, là, ce sont parmi nos meilleurs indicateurs de la santé écosystémique de nos lacs. Je vais terminer avec une série d'images qui va vous donner une idée de ce qu'on fait au Parc de la Gatineau, de ce qu'on a fait dans le passé et de ce qu'on fait encore présentement. Parlons un peu du lac Myes. Encore une fois, il n'y a pas de moules zébrées qui ont déjà été observées au Parc de la Gatineau et c'est une très bonne nouvelle. On se croise les doigts. Il faut insister de ne pas apporter de bateaux à moteur dans ces lacs-là. Pas de rampe à bateau avec des bateaux qui font le va-et-vient. Donc, on a des barrières. La gestion du Parc de la Gatineau est consciente. Il ne faut pas introduire la moule zébrée dans le Parc de la Gatineau. Ça serait un peu dévastateur, merci. Le lac Myes, dans le passé, au début des années 2000, on a fait tout le tour du lac avec des étudiants. On avait une trentaine de sites. Vous les voyez ici, là, sont tous numérotés. On a la densité de moules d'eau douce, la diversité. Donc, on a fait les grands lacs principaux du parc. On a utilisé une méthode du quadrat, ici qu'on voit, du tuyau de PVC, et 1 mètre par 1 mètre, une tige qui indique la profondeur verticale, une poignée pour le prendre si tu es en wetsuit, et c'est une méthode qui permet de quantifier les moules d'eau douce. With this device, you can just put it on the bottom, and with a wetsuit, you go, you check with your mass, shallow water, deep to about a meter, meter and a half. You can count the mussels, determine the diversity, and along transects, and study the perimeter of the lake. The team that we had at the time, students, research assistant, and so, you know, to do this work, you depend on a lot of hand power, a lot of people work, volunteers, students, and together you produce, you know, these kinds of data set. So the elliptio, the most common species in the park, for each of the sites, the density per square meter. I just want to mention something here. If you look at this figure here, you can see that the discharge area of Lac Myes, what we call Small Myes Lake, where there is this little bridge crossing along the trail number 1. Look at the density of freshwater mussels. It is superior there compared to everywhere else along Myes Lake. One of the reasons, habitat, shelter for small-bodied fishes, most probably, large woody debris, sunken trees, fallen trees. That portion of Myes Lake is the wilder one, a bit like the Northern Basin. That allows minnows, sticklebacks to hide, to find shelter from bass predation, which is good for mussels. So the less predation on the small fishes, the more reproduction of our native mussels. So the bass can be a problem there. But thanks to large woody debris, fallen trees, you've got more microhabitat, more shelter. Here's a picture of a crayfish here that I took. Lac La Pêche, I will just skip that very quickly. We did the same at Lac La Pêche. Different species there. We even have the fat market in Lac La Pêche. Lac Philippe, we did the same. I will mention something about Lac Philippe right here at the discharge on the way to Lac Mousseau. La décharge du lac Philippe est intéressante. Il y a une espèce particulièrement abondante qu'on appelle l'alaspidonte à fortes dents. The triangle floater is common in Lac Philippe, which is not the case in Lac Myes. and this one is found right here. And we think it's because there are some minnow species. Some minnow species traveling along that stream that connects Lac Philippe to Lac Mousseau. And these minnow species are diverse. There are many of them, and they allow the reproduction of this particular mussel. So that's Lac Philippe for you. And at the end, we've done the same thing. I could show you histograms, but that's not the point. I want not too much data is needed at this point. But to mention again that the discharge of Lac Philippe is particularly interesting. La décharge du Lac Philippe, vous la voyez ici, vous reconnaissez ce secteur. On peut aller, les cavernes lorsqu'il sont ici, lorsqu'il cave. Alors, cette plage-là, bien connue de bien des gens, bien des canoteurs, est un endroit où ces moules-là se retrouvent. Il y a 6 moules au mètre carré et celle-là, elle est commune. C'est un site particulièrement intéressant. Une photo résumée un petit peu. Vous voyez sur cette photo-là les espèces jusqu'à ce jour qu'on a retrouvées dans les ruisseaux, rivières et lacs du parc de Gatineau, surtout les lacs. Il nous reste beaucoup de travail à faire dans les ruisseaux, la rivière, la pêche, j'en parlerai dans une minute. Mais vous voyez par ordre d'abondance. La plus abondante, c'est l'elliptio de l'Est. La moins abondante, la plus rare, la numéro 8, c'est la lasbigone des ruisseaux. Retrouvée uniquement dans un seul ruisseau, dans les années 1950, je n'ai pas encore confirmé qu'elle est présente à ce jour. Mais on a un spécimen dans nos collections nationales, prélevé entre le lac Mousseau et le lac Philippe, et qui montre un spécimen de cette espèce-là. C'est vraiment une moule très rare au parc. Est-elle encore là, cette population? Je soulève la question. Bref, Eastern Elliptio, giant floater, and there's a second species, by the way, of the genus Piganodon or Lanodon. We have the genome for it. We're not too sure what it is, but it's a different species. So I could add even a species right there. Triangle floater, the fat market, the eastern lac mussel, the creeper coming in Lac Taylor, a bit in Meech Lake, and in also Meech Creek. One specimen found by a volunteer. The cylindrical floater, a rare animal, and this one is very rare. Future research before I wrap up. The areas for which we know the least by far, I have yet to really spend time myself, my team, volunteers, keen volunteers. We work with keen volunteers in Chelsea and elsewhere, and that's true for Gatineau Park. The streams and the Rivière La Pêche itself. We've got work to do. This is a typical picture of Chelsea Creek, the ruisseau Chelsea, and thanks to volunteer of the Acre group, on parle ici de l'organisation Chelsea pour le respect de l'environnement. Donc, Acre, this is the association for the protection, the respect for the natural environment that you have in our area. Acre. So, thanks to Stephen Woodley, Scott Finlay, we were putting together, it started, we began last fall, and this is an example of the Chelsea Creek where, and here's my son Julien, who not too many years ago discovered the first mussels of the creek, and I was there wondering if there were mussels there. Indeed they are, and thanks to volunteers, I could mention a few names there, but in Chelsea, a number of keen volunteers came with us and they helped me, and they did map. Thanks to their effort, we started last year and we will continue next year, some of the mussel sites along that creek. So here's an example of this anodonte cylindrique, the cylindrical floater mussel, and that's the fish that we believe is the host needed for the reproduction of this particular species. I have to mention something. Chelsea Creek, le ruisseau Chelsea, il y a énormément de cyprins, de dards, beaucoup de petits poissons, des bancs de poissons partout. C'est un ruisseau riche pour les poissons, mais jusqu'à date, on n'a retrouvé qu'une seule espèce de moule d'eau douce. So a lot of small-bodied fishes in Chelsea Creek, minnows, cyprinids, and this one is the Iowa darter, and we believe it's the one fish that is at least partly responsible, possibly several species as well as this one, responsible for the reproduction and dispersal of the cylindrical floater mussel. So future research, again, wrapping this up, the fish-mussel relationship for all of Gatineau Park freshwater mussels is poorly known. It's also true for the entire Ottawa River watershed. It's also true for many species in Canada. It's difficult to study that. You need DNA, you need to extract these tiny glochidia on the gills behind the operculum here or on the fins. Do the DNA confirm which mussel you're talking about to confirm accurate connections between, let's say, this fish and the mussel, this one and the mussel, or what. So there's a lot of species to look at. It's a difficult work. That's one of the things we're interested in the coming years, the fish-mussel connection. So again, when I take my camera underwater, that's what I see. Without disturbing them, I don't need to even touch them sometimes. I just look at the siphons. I look at the siphon apertures and I can tell what species I'm dealing with. So I encourage the people today in this audience to pay attention to the appearance of undisturbed mussels as they filter feed. And if you want, if you've got a great picture, send me the picture. I will try to help you to identify them. And again, to raise that question, what is the exact fish mussel connection for each of those? So au parc, quels sont les poissons nécessaires à la reproduction de ces différentes espèces de moules-là? This is a black box. It requires years of work, and we are pursuing this. It's a valuable endeavor, and stay tuned for the coming years. I just wanted to maybe read this one here in terms of the service that they provide. Really, think about it. À l'intérieur de cette coquille, de cette moule vivante, que le temps a couvert d'algues et de sédiments. This animal is old here, folks, maybe 10, 20 years old. Il y a un animal qui vit, qui a un cœur qui bat, un animal qui oxygène le fond du lac ou de la rivière avec son pied mobile et dont les branchies filtrent l'eau de la rivière 24 heures par jour, 7 jours sur 7 pendant plusieurs mois de l'année. Parfois, ça peut aller jusqu'à 8 mois. Et ça pendant des décennies parce que ça vit des années des moules d'eau douce. Et là, je mentionne ici, j'écris « Quel grand service écologique ». Alors, lors de votre prochaine sortie en canot ou au parc de la Gatineau, le premier but de ma présentation, le but numéro 1, and I hope that I touch base on that, lors de votre prochaine sortie, c'est de voir les moules d'eau douce sous un angle différent. Maintenant que vous avez une meilleure perception de la connectivité, le lien poisson-moule pour chaque espèce qui est bien particulier, tel poisson, tel moule, combinaison de poissons, etc. I hope that with these images that you've seen, the biology that I have covered, you appreciate further this amazing group of animals, silent, but playing an amazing ecosystem service to us, to our lakes and rivers. I appreciate your patience. I'm done. Quentin, thank you so much for overseeing this presentation.